Water. This is a repetition of last week's class. So let's look at water and all the properties of water, which are important not only from the food science and food safety point of view, but from all angles. What is water and what are the properties of water? So, if you look at water, water is a very simple molecule which is made up of two hydrogen atoms, which is bonded to one oxygen atom. And it is not a symmetrical molecule because the hydrogen are not symmetrical. And you will see that the electrons, which in oxygen spend more time, the electrons spend more time near oxygen and less time near hydrogen. And so, therefore, the oxygen is electronegative, whereas the hydrogen will be electropositive, and this gives water its polar nature. So, water is the only substance on Earth which is found in all the three states. It occurs as liquid, as solid, and as a gas, that is steam and vapor. So, when water molecules move very fast, they tend to break their hydrogen bonds, as we will see, and it will cause evaporation. However, when the gas or the vapor molecules slow down, the hydrogen bonds will be formed, they will join together, and condensation occurs. So, the density of water decreases with temperature. The properties of water: at sea level, pure water boils at 100 degrees. It freezes at 0 degrees. The boiling point of water decreases at higher elevation. That means as you move up to the hills, you will see that it will take a longer time for water to boil. And water boils faster at sea level. For this reason also you will see cooking of vegetables, boiling of an egg, etc. take longer at higher altitudes where the pressure is less. So the properties of water: we know that water is a polar molecule. It will exhibit cohesion and adhesion. It has a very high specific heat. The density of water is greatest at 4 degrees centigrade. It is a universal solvent of life, and also we will discuss osmosis. So now looking at the water molecule, as we say, it is polar. It exhibits polar properties. That is because the electrons, the concentration of the electrons is more near the oxygen and less near the hydrogen. So the hydrogen tends to be positive and the oxygen end of it tends to be negative. However, the net charge of water will be zero, because you will see that the two negative of oxygen are neutralized by one proton each of the hydrogen. Because of this nature of it being both positive and negative, water is known to be dipolar. What is shown on the right is the Bohr model of water. So now coming to the water molecule, you look at the water molecule. These water molecules are attracted to one another because one, water is dipolar, so the oxygen is negative, the hydrogen is positive. So the positive end of hydrogen will be attracted to the negative end of another water molecule, and the oxygen end will be attracted to the hydrogen of the water molecule. So in a liquid, in the water liquid, the water molecules are held together by what are called as hydrogen bonds. That is a bond between oxygen and hydrogen. So the hydrogen bonds exist between water molecules. One would ask, hydrogen bonds are very weak, so how is it possible that water still exists as a liquid? One, one hydrogen bond is weak, but in a water molecule, you will see that thousands of water molecules, each molecule will be hydrogen bonded to four of its surrounding neighbors. And so the large number of hydrogen bonds will make them strong. So the hydrogen bond occurs between the positive of H plus with the O minus of the oxygen. So as I said, each water molecule can hydrogen bond with four neighbors. That means with four water molecules around it. So this is a single water molecule. So you see it has hydrogen bonded with this. This hydrogen can hydrogen bond with this. The oxygen has two, so it can hydrogen bond with this hydrogen and with this water molecule and with this water molecule. So therefore, a single water molecule can have four hydrogen bonds, and a large number of hydrogen bonds put together makes them very strong. Individual hydrogen bonds are weak bonds compared to ionic bonds or covalent bonds. So all the properties of water, that is the unique properties of water, such as the cohesive behavior, the resistance in changes to temperature, the very high heat of vaporization, that is 540 calories per gram, its expansion when it freezes, and it acting as a versatile solvent can be explained on the basis of the hydrogen bond. So what is cohesion? Cohesion is attraction between particles of the same substance. So you know why water is attracted to itself, because of the hydrogen bond between the oxygen and the hydrogen. And this cohesion or attraction between the water molecules results in what is known as surface tension. That is a measure of the strength of the water's surface. And this surface tension produces a surface film, that is a very strong film because of the hydrogen bonds, which cannot be broken. And this allows for insects and other small animals to walk on the surface of water. So surface tension. What is surface tension? Surface tension is a measure of the force that is required to break the stretch of the liquid, that is related to cohesion. So because of cohesion, the water molecules are held very tightly, and so you will need a large force to break those hydrogen bonds and make it dissociate. Water has a greater surface tension than other liquids. Of course, it is due to the number of hydrogen bonds on it. It behaves as if covered by an invisible film, and some insects, like what you see as the water boatman or the other, you will see that they can run or walk on it. However, humans cannot walk or run on water, and please do not try this, because you have to only swim, you cannot walk on water. So next you have adhesion, which is exactly the opposite of cohesion. Cohesion is an attraction between similar molecules. So it is the attraction of water molecules between themselves. Adhesion, on the other hand, is the opposite. It is attraction between two different substances. So water molecules can get attracted or form hydrogen bonds with other surfaces such as glass, the silica in glass. It can form hydrogen bonds with the soil. It can form hydrogen bonds with the plant tissue as well as with cotton. And it is because of this property of adhesion, what you will see is the capillary action of water molecules, which pull along or tow along one molecule behind the other. And so you can see the rise of water in a thin capillary glass tube. This property of adhesion also can explain the process of transpiration. So how do leaves transpire? You know that leaves will give out water vapor, and how do they get this water? They can remove, the trees and plants can remove the water from the soil by this capillary action. You also see that the filter paper or paper towels that you use soak up water. So what is this due to? This again is due to the properties of adhesion, which lead to capillary action. Also, in the lab, you will see that your mobile face in paper chromatography or thin layer chromatography, you don't need to apply a force. The water or the mobile phase is easily taken up, or it moves up along the paper or the thin layer due to the capillary action. And this capillary action is due to adhesion, that is the attraction of water to the molecules in either the paper or the thin layer or the paper towel. So adhesion causes capillary action, and the movement of water within the porous material of of any paper or the thin layer is due to three properties, that is surface tension, adhesion, and cohesion. So the water molecules will be attracted to each other by cohesion. So they will form a thin layer. On the other hand, the water molecules will be attracted to glass, that is the silica, SiO2, and it will be there will be adhesion, and the surface tension, that means the thin layer is formed. So these three properties put together can explain why water moves up plants or trees against gravity. So if you want to lift water, if you have noticed, if you want to lift water from a well or from your sump, you cannot lift it by, it cannot move up due to gravity. You have to use some external force such as a pump. But on the other hand, have you ever wondered why, how plants will take up the water against gravity? So cohesion is responsible for the transport of water in columns in the plant. So the cohesion among water molecules plays a key role in the movement of water against gravity, and adhesion, clinging of the water molecule to the sides of the plant contributes, is another contribution, as the water will adhere to the walls, that is the cell wall of the plant. And so therefore you see that the nutrients also, we add to the base of the plant, and the nutrients are taken up by the xylem and the phloem, that is the liquid in the plant. So addition also, if you have looked at nature, is responsible for these small dewdrops or spheres that you see early morning on plant leaves or on flower petals. You see them, it is due to adhesion. Another beauty that you see is the attachment of water molecules to a spider web. So this again is due to the attraction of water molecules to the silk threads, that is the silk threads on the spider web. So cohesion and adhesion are two opposite forces. Cohesion is the attraction between water molecules to each other, and adhesion is the attraction of water molecules to another substance. In the laboratory, cohesion also plays a role to explain the meniscus that you see in the glassware, especially when you're using a pipette or when you're using a burette. You will see that whenever you use water or buffer, you will see that it forms a concave meniscus. And the bottom of the meniscus is used to indicate the volume of the liquid. So When you are measuring the volume of a liquid, always keep your eye at level with the meniscus and read the bottom of the meniscus in case of a pipette or a burette for aqueous liquids. However, on the other hand, when the cohesion or the attraction between the molecules is greater than the adhesion, like for example mercury, where the attraction between the mercury molecules is far higher than between mercury and glass, what you see is a convex meniscus. So if you have a thermometer at home, please look at the meniscus of the thermometer and see whether you can see a concave meniscus, or if you go back to the laboratory, check whether you have a concave meniscus. So a concave meniscus is formed when the attraction between the molecules, that is the cohesion between the molecules, is greater than the addition to the glass surface. So now let's come to another property of water. Water is known to have a very high specific heat. So what is specific heat? It is the amount of heat required to raise or lower the temperature of 1 gram of water by 1 degree centigrade. So you will find that water resists temperature change both for heating and cooling. Water can absorb large amounts of heat energy with a very little change in the actual temperature. So if you look at water when you want to do cooling, you will find that when you add cold water, something hot into cold water, the temperature of the water does not change much. That is what is said by the high specific heat. And this is very important from the nature's point of view, because is very important from the nature's point of view, because it prevents the fluctuations in the temperature uh, and the outside temperature, and this is the reason that we have colder, I mean milder climates in the um, coastal areas. So the water in the coastal areas has the property of absorbing the heat. This high specific heat of water is also responsible for the stable marine environment. If water heated very fast, then the marine animals would not survive. Aquatic organisms face less temperature variation than terrestrial organisms. And also, because of the high specific heat of water, you will find that water is used in cooling systems which are found in cars and in industries. And in the laboratory also, you will see that in your condensers, you will be passing water, because the water can absorb the heat without much change in the temperature. So coming to the temperatures on the earth, you will see that water stabilizes the air temperatures by absorbing heat from warmer air and releasing heat to cooler air. Water also absorbs relatively large amounts of heat with only a slight change in its own temperature. That is because of the high specific heat. So if you look at the temperatures of water, water boils at 100 degrees, human body temperature is 37 degrees Celsius, room temperature is 23 plus or minus 2 degrees Celsius, and the freezing point of water is 0 degrees Celsius. Next property of water is that it has a very high heat of vaporization. What is heat of vaporization? It is the amount of heat energy required to convert 1 gram of water into a liquid. So in order for water to evaporate, as you saw in the previous slides, for evaporation to occur, the hydrogen bonds must break. So when evaporation occurs, hydrogen bonds, that is, the water molecules are moving fast, the hydrogen bonds are broken, and it evaporates. And during evaporation, along with the hydrogen bonds being broken, water will take heat along with it, and it will cause cooling. So water's heat of vaporization is 540 calories per gram. In order for water to evaporate, each gram of water must absorb 540 calories of heat. As water evaporates, it removes a lot of heat with it, and that is what you see as the cooling effect. So evaporative cooling, the cooling of a surface occurs when liquid evaporates, and you will see that when you sweat, because the water is evaporating, actually your body temperature is coming down. And this evaporative cooling property of water is responsible for moderating the Earth's climate, for stabilizing the temperature in the aquatic ecosystem, and from preventing organisms from overheating. So it is now summer in the south of thing, where we are experiencing very high temperatures ranging from 37 to 40 degrees Celsius. So if we have to bear this heat, the body, and if the body could not sweat, then you will see that the body temperature would have increased. That is the surface temperature. However, the sweating resorts to evaporative cooling, and so therefore you see the surface temperature of your body is always maintained. Water is less dense as a solid, and this is easily visible from the fact that ice will float in water. So liquid water has got hydrogen bonds that are constantly being formed and broken and reformed. So in frozen water, you will see that there is a crystal-like lattice where the molecules, the water molecules, are set as a fixed distance. Because there is a large amount of space between each of the water molecules, the ice will float. So the density of ice will go down because it has got these large spaces. And this floating of ice is also very helpful for the aquatic organisms, because the ice will form on top, and it is less dense than water. It will float, and lakes will freeze actually from the bottom, and it would prevent the lakes from freezing and killing the aquatic environment. So this is the structure of two water molecules. So from these molecules, you should be able to tell which is water and which is ice. So in water, I said the molecules are compact and there will be no space, whereas in ice, there will be space. So this is water and this is ice, and the space in the crystal lattice of ice causes the ice to be less dense and float. So this is water, and on the right hand side is ice. So looking at the density of water, the density of water is highest at 4 degrees centigrade. It contracts until it reaches 4 degrees centigrade, and then it expands from 4 degrees to 0 degrees. So this density of water, the higher the density at 4 degrees, also is responsible for preventing water from freezing from bottom up. Ice forms first on the surface, and it makes transition between seasons less abrupt. Now coming to a property that all of you are very familiar with, and that is using water as a solvent. Water is known as the universal solvent because it can dissolve almost all ionic compounds. So in a solution, you have two components. One is the solute, which dissolves in the solvent. So when the solvent is water, the solution formed is known as an aqueous solution. And water dissolves hydrophilic compounds. All compounds that are dissolved in water are hydrophilic, and these hydrophilic compounds are generally ionic compounds. That means those which can dissociate into positive and negative charge, and those that are polar molecules. Water does not dissolve hydrophobic compounds, that is the non-polar compounds such as hexane, oil, edible cooking oil. If ever you have tried to mix oil and water, you will see that oil floats on top. So the terms that you need to know and understand are solution, aqueous solution, what is hydrophilic, and what is hydrophobic. So in solutions, you have two types of solutions. One is a solution and the other is a suspension. A solution is a homogeneous mixture wherein you cannot see the difference between the solute and the solvent. So it will be completely mixed. And a suspension is a non-homogeneous solution is known as a suspension. Like for example, sand and water. But however, some suspensions you will see as homogeneous because they have used what are known as emulsifiers to mix the solute, that is the solvent, the immiscible solvent and solute, as in the case of milk and mayonnaise. So in a solution, ionic compounds and polar compounds disperse very easily. They are evenly distributed and they are called homogeneous. The solute is generally the substance that is being dissolved. The solvent is generally the substance in which the solute dissolves, or the one which is in higher quantity. So water as a solvent, as I said earlier, is a universal solvent. The properties of water as a universal solvent include that it interacts with polar compounds, so like sodium chloride, sodium carbonate, sodium bicarbonate, potassium chloride. So all these are polar compounds and can form solutions. It is repelled by non-polar compounds. The third property is the small size of water allows it to saturate the areas. And it is this saturation that makes the polar compounds go into solution. And it can convey other substances in solution. So water as a solvent. Now, if you take some salt, some sodium chloride, and you put it into water, what do you see? The sodium chloride dissolves and you get a solution. You get a salt solution. But the chemistry that takes place is, if you look at when you put salt into water, the salt molecule attracts a large number of water molecules, and these small water molecules which are attracted to the sodium chloride molecules break the ionic bonds, or it causes the salt crystals to fall apart. So the strongly charged salt ions are attracted to the weakly charged water molecules. The large number of water molecules surround each salt ion, and then the salt crystal falls apart or dissociates and it dissolves. So a solution is formed. So this also explains water is especially good at dissolving salts. So salts form from the combination of particles with opposite electrical charges. Suspensions, they do not dissolve completely, but they separate. However, you can make a homogeneous suspension, as I said, by using emulsifiers. Milk is a suspension. of fat particles in water, which is in suspension due to the protein molecules which act as emulsifier. So let's come and look at the dissociation of water molecules. One water molecule in 550 million can dissociate to give you a hydrogen, proton, and a hydroxide ion. So water can dissociate into a hydrogen ion and a hydroxyl ion. When you have two water molecules, you have the proton is taken up by this water molecule to give you a hydronium ion and a hydroxyl ion. At room temperature, that is 23 plus or minus 2 degrees centigrade, in pure water, the concentration of the hydrogen ions is equal to the concentration of the OH ions, and they are each equal to 1 into 10 to the power of minus 7 molar. So you have 1 into 10 to the power of minus 7 molar hydrogen ions, and you have 1 into 10 to the power of minus 7 hydroxide ions. So the hydrogen ions, the dissociation of water into a H plus or a proton gives it the property of being an acid. So we define acids by a term which is known as pH, or pH is the negative logarithm to the base 10 of the hydrogen ion concentration. So always the negative logarithm to the base 10 is represented by P, and it is the negative logarithm to the base 10 of the hydrogen ion concentration in moles per liter. So every time you want to calculate a pH, please see that the hydrogen ion concentration is in moles per liter. Many a times I see people writing pH in the wrong way. Please remember, it must always be written with a lowercase p and an uppercase h. And this definition of negative logarithm to the base 10 was introduced in 1909 by the Danish biochemist Sorensen. So in a neutral solution, or in a solution, as I said, the concentration of the hydrogen ions is equal to the concentration of the hydroxyl ions, and each of them is equal to 1 into 10 to the power of minus 7 molar. So now let us look at what will be the pH. So we have pH is negative logarithm to the base 10 of the hydrogen ion concentration. So this is equal to log of 1 plus log into 10 to the power of minus 7, and so this gives you a pH of 7. So the hydrogen ion concentration and hydroxyl ion concentration are related by this term, this plus thing into this. So you have 10 to the power of minus 14 is the concentration of the water. Now in neutral solution, we, as I said, H plus will be equal to OH minus, and we know that H plus is 1 into 10 to the power of minus 7, and the pH is 7. Now let us see the same thing with what will be the negative logarithm to the base 10 of the hydroxyl ion, where the concentration is 10 to the power of minus 7 moles per liter. So you will see it is exactly the same. So now pH plus POH will be equal to 7 plus 7, or 14. This is a relationship that you must remember, that pH plus POH is equal to 14, whereas H plus into OH minus, the hydrogen concentration, is equal to 10 to the power of minus 14 moles per liter squared. So let's come to the pH, and what is this? So what is alkalinity and what is acidity? It is the measurement of the hydrogen ion concentration which is found in the particular substance. The scale goes from 0 to 14, and now I'm sure all of you know why it goes from 0 to 14, because 0 to 7 is due to the hydrogen ion, and 7 to 14 is due to the hydroxyl ion. 7 is neutral, below 7 is acidic, above 7 is alkaline or basic. And 1 pH unit represents a 10-fold change in the hydrogen ion concentration, because you know that it is minus negative logarithm to the base 10. That is a 10-fold change. So therefore, if you look at this, so if you have a pH of 3, it is 1000, it is 10 to the power of 3 times stronger than pH 6. pH 4 will be only 10 times stronger than 3. pH 5 will be 100 times stronger than 3. And pH 2 will be 10 times stronger than 3. So it is, every pH unit represents a 10-fold change in the hydrogen ion concentration. So pH and pOH are related by this table, where pH plus pOH is equal to 14. So if the pH is equal to 2, pOH will be equal to 12. If pOH is equal to 11, pH will be equal to 3. So this table just gives you an idea of the same relationship. So if you're given the pH of a solution, you should be able to calculate the pOH. If you're given the pOH of a solution, you should be able to calculate the hydrogen ion concentration. So acids, they are strong. So let us look at what are the acids in our normal life, everyday life. So you see, human blood is about pH 7.4. Pure water is pH 7. Milk is about 6.8. So normal rainfall, acid rainfall. Tomato juice has a pH of 4. Lemon juice has a pH of 2.5. And acid, your stomach acid, has a pH of 1.5. So usually that is why you talk about acidity in the stomach. Let us look at some bases. So the bases that you use, your oven cleaner, the bleach, or the ammonia solution, are very strong bases because their pH is in the order of 12 to 14. Soap also is alkaline. You have a pH of 10. Seawater is alkaline. So because of the alkaline nature of soap and bleach, when you use the surf or these soaps to clean your glassware, you will see that your glassware will be alkaline. So therefore, it is better not to wash your glassware with surf or with liquid soap, but to wash with soap solutions which are neutral in pH. A large number of soap solutions such as Laboline and neutral soap solutions are available. Please make sure that you use the neutral soap solutions to wash your glassware. Otherwise, the pH of the glassware will interfere with the analysis. The blood has a pH of 7.3, average is 7.4. It's between 7.35 and 7.45. And this pH is maintained by the buffers in the blood, such as the bicarbonate buffer. Now let us see some examples of how to convert pH to hydrogen ion concentration and hydrogen ion concentration intake. The first problem is convert 0.0025 molar hydrochloric acid into pH. Hydrochloric acid is a strong acid and it is completely ionized. So you have pH is negative logarithm to the base 10 of 0.0025. And this log of 0.0025 is minus 2.6. So minus into minus is plus. This is very easy for you because in your calculators or in the calculator on the computer, you just punch in this number and ask it to give you the log. Now, what is the hydrogen ion, vice versa? What is the hydrogen ion concentration of a solution which has a pH of 8.34. So again, applying the formula, you have 8.34 is equal to minus log to the base 10 of the hydrogen ion concentration. How do you convert this? Actually, the hydrogen ion concentration is the anti-log of this, or the hydrogen ion concentration is 10 to the power of minus 8.34, which is 4.57 into 10 to the power of minus 9 molar. Now you will ask me in a calculator, because now most of you have forgotten how to use the Clark's tables, that is the logarithmic and the anti-log tables, and we are so dependent on the calculator. So to calculate 10 to the power of minus 8.34 in your calculator, either in the computer or the calculator, punch in minus 8.34 and then punch in inverse log, which will give you the anti-log of this, and it will give you a value of 4.57 into 10 to the power of minus 9 molar. So similarly, you can do calculations of POH if you're given the OH concentration. The relationship between pH and POH is 14. So in this problem, if the solution has a POH of 11.6, what is the pH of this solution? Very simple. 14 minus POH, which gives you 2.24. So for the food analyst exam, you should be able to convert pH into hydrogen ion concentration into pH. If you're given POH, you should be able to calculate pH. If you're given POH, you should be able to calculate the hydrogen ion concentration. So here is a different type of a problem, but again, which involves the POH and the pH, but the question is put forward in a different way. So if 4 grams of pure sodium hydroxide is dissolved in 10 liters of water, what is the pH of the solution? So the solution is, you first need to calculate the hydroxyl ion concentration. So how do you calculate this hydroxyl ion concentration? Now take the sodium hydroxide. Do you know that the molecular weight of sodium hydroxide is 23 plus 16 plus 1, which is equal to 40. So 4 grams is equal to 0.1 mole. So you have 4 grams of sodium hydroxide, or you have 0.1 mole of sodium hydroxide in 10 liters of water, or that is the same as 0.01 mole per liter. So the concentration of sodium hydroxide is 1 into 10 to the power of minus 2 moles per liter. Sodium hydroxide is a very strong base and can dissociate into Na plus and OH minus. So 1 mole of sodium hydroxide will give you 1 mole of sodium and 1 mole of hydroxyl ion. So therefore the hydroxyl ion concentration is 1 into 10 to the power of minus 2. So the POH will be equal to 2. Okay, it's minus logarithm to the base 10, and we know that pH plus POH is equal to 14. So therefore the pH is equal to 12. So you should read the question carefully, because many a times we may not ask you the POH, we'll ask you the pH, or we will ask you the hydrogen ion concentration. So these problems that I have shown you are just representative type of problems that you could get in the food analyst exam. Now, water also participates in a number of chemical reactions. It is a reactant. So it is involved in dehydration, where a water molecule can be removed and a bond is formed, called a dehydration. Or it could be a reactant where it is used to lyse a bond or break a bond, or hydrolysis. So water can participate in dehydration reactions as well as in lyase or hydrolysis reactions. So let's come to what you should know about the standards of drinking water. Drinking water is also known as potable water or is defined as water that is safe to drink. According to WHO, safe drinking water is defined as that which does not represent any significant risk to the health over a lifetime of consumption, including different sensitivities that may occur between life stages. So, access to safe drinking water is essential to health and it is a basic human right. So, what are the standards for drinking water? So, the standards for drinking water are defined by certain properties. That is, we have the physical properties, it should be clear, it should be free of suspended matter, it should be colorless. We have the so physical and chemical properties include heavy metals, trace metals, total suspended solids, hardness, turbidity, taste. All these come under physical and chemical and organoleptic parameters. The microbiology parameters, one of the most important microbiology parameters in drinking water is that it should not contain coliform bacteria, it should not contain E. coli, it should not contain viruses, protozoans, it should not contain any organisms which can cause disease. The chemical parameters pose a problem because by just drinking a small amount doesn't cause any disease. But however, buildup of this means if you continuously drink water which is laced with lead, you will end up with lead, some repercussions due to lead. So if you look at our standards, FSSAI does not have a standard for drinking water. FSSAI has standards for mineral water and for packaged drinking water other than mineral water. However, for drinking water means this is for water other than bottled water. You will see that we have an Indian standard which goes by IS 1050002012. So all drinking water should meet this Indian standard. All water that is used in food manufacture by the FBO should meet the drinking water standard. So water in the laboratory. So which water do you use in the laboratory? Do you use drinking water or do you use drinking water standard or you use the bottled water standard? No, there are different standards for water that is used in the laboratory because this is a common thing. And often we overlook which grade of water is being used. It is essential that for every analytical measure, you see the water that is recommended, the grade of the water that is recommended. So if you look at the Indian standard, we have IS 1070-1992, which is reaffirmed in 2008. It says standards for reagent grade water. So we have grade 1, grade 2, and grade 3, which specifies what it should be. So which of these grades will you use? So if you look at grade 1, it requires minimum interference and it is used in tests which require minimum interference and maximum precision and accuracy. So where will you use this water? You will use this water in LCMS-MS, GCMS-MS, ICP-MS, where even minute quantities will be there. Then you have grade 2 water, which is like your distilled water, and your double distilled or glass distilled water, which is used for buffers, making pH solutions, for dilutions, for your volumetric and your gravimetric analysis. And grade 3 water is the water which can be used to wash glassware, the preliminary rinsing of glassware, and final rinsing of glassware is through grade 2 water. This grade 3 water can be used in autoclaves, in water bath, in feed water for making higher grade water, and in the condensers. The ASTMR or ASTM also has given standards. This is by the American Society. But here, instead of grade 1, grade 2, and grade 3, you have type 1, type 2, type 3, and type 4. So here, the single asterisk shows that you should actually filter it through a 0.2 micron membrane filter. Type 2 is prepared by distillation, and type 3 requires the use of a 0.45 micron. So now you look at the parameters. All the parameters are given. So you see the silica content of type 1 has to be very low. So type 1 is very stringent, and it is the water that is used. So type 3 is like our grade 3. It is recommended for glassware rinsing, and so type 2 is like our grade 2 for general laboratory preparation, buffers, pH solutions, etc. And type 1 water is where you use it for the very sensitive analytical measurements such as GC, HPLC, GCMS, MS, amino acid analysis, ICP-MS, in molecular biology, experiments for DNA sequencing, PCR, etc. And of course, type 1 is more expensive to produce than 2 or 3. Similarly, grade 1 water is more expensive to produce than grade 2 or grade 3. So now let's come to the last part of this class, where the importance of water in food. So water is an essential constituent of food, and it can occur as an intracellular or an extracellular component in animal and vegetable foods. It is the dispersing medium or solvent in many foods, and it is a minor constituent in some foods, but in some foods it is a major constituent. For example, in milk, it is the major constituent. In apple, it is the major constituent. In orange, it is the major constituent. However, if you take nuts such as almond or peanut, you will find that water is a minor constituent. There are two terms which describe water in food. One is moisture, which refers to the total amount of water present in the food, and the other is water activity, which refers to the form in which water exists. These two are entirely different. Please do not think moisture is the same as water activity. So look at the water content of some selected foods. So you have water content of apple, orange, milk being almost 87%. Water content of tomato, lettuce, and cabbage is very high, whereas the water content of milk powder, rice, wheat flour are much lower. So the amount of water is only about 4%. And some products contain only 0 to 2% of water. So what are the forms of water in food? In food, water exists as in three forms. One is the free water. Then you have adsorbed water, and then you have bound water. So water can exist as free water, adsorbed water, or bound water. Chemically, each form of water is the same. Whether it is free water, adsorbed water, or bound water, it is H2O. The only thing is it exists in different physical and chemical conditions. So the form in which the water exists, the free water, adsorbed water, or the bound water, actually determines the physical properties of the food and also determines the shelf life, how long the food can be kept. Now, if you go back, we saw that the water content of apple and milk are the same, 87%. But you see the physical form. In apple, it is a liquid, and it is in milk, it is a liquid. There is 87%. Whereas the same thing in an apple, the water is absorbed to different molecules and things, but they have the amount of water is exactly the same, but they have different physical structures. So you have liquid milk and you have the apple. So free water. What is free water? Most water in foods is called free water. It's lightly entrapped and therefore it's easily pressable. So for example, you can make apple juice, you can make orange juice, that's the free water. So all the free water will come out and it acts as a dispersing agent, and free water can easily be removed and you can make it dry. So if you dry milk, you get milk powder because you have removed all the water. Adsorbed water is the water which is associated in a layer because of its hydrogen bonds, and it surrounds the hydrophilic molecules in the food, especially the protein, and it is held between the cell walls. So you have the adsorbed water. And bound water is the water that remains unfrozen at temperatures below 0 degrees. This bound water will not be available for any of the reactions as a solvent. It is bound to the different components. The unfreezable water is based on the protein content and varies. So it's about 8 to 10% in animal tissue. Egg white contains about 0.4 grams of the bound water. Most fruits and vegetables contain less than 6% of unfreezable water. In fruits and vegetables, most of the water is the free water, that is water that can easily be squeezed out, or it could be pressed out of the fruit or vegetable. So how do you determine moisture content? Moisture content is determined by several methods, and I'm sure all of you know that it is one of the most basic methods is the hot air oven drying method, where you use the pans and you use a convection oven, you pre-weigh your sample, then you dry it in the oven till you get a constant weight. So, what are the factors which affect the moisture content? This is the convection oven, the particle size. You will find that if the particle size is very big, it doesn't dry. The moisture can be held between the particles. So, if you break it down into a smaller particle size, you will see that a smaller particle size and the surface area affects the rate of loss. At very high temperature, the dryness is complete, but however, you have to be very careful at what temperature you dry, because you don't want it to char, or you don't want it to decompose, or you don't want it to lose the volatiles. So the temperature generally that we use is 105 degrees or 133 degrees. So in the FSSAI regulation, you will see moisture so many percent with oven method at 105 or oven method at 135. Oven drying takes a long time for the water to be removed, and because of that, for the sensitive foods which are destroyed by exposing to high temperature for very long time, we use the vacuum oven. That means you do the drying in the presence of vacuum at a lower temperature and you get it. So the two oven drying methods are the hot air and the vacuum oven method. We use the distillation method or the Karl Fischer titration for low moisture foods. So in the distillation method, especially foods which have got less than 2% moisture, it's very difficult to get the moisture content. So what is done in the distillation method is that the substance or the test substance is mixed with an organic solvent which has a higher boiling point than water but is immiscible with water. The solvents we use are generally toluene, xylene, or benzene. So here the sample along with the solvent is put into the flask. This flask is not directly heated. It is heated in a water bath or in a sand bath, and the water will boil along with the solvent and it will evaporate, and with the help of this condenser both the solvent and the water will condense in this condenser, and the volume of water can directly be read off. So water is measured directly in the distillation procedure, but the reading of volume of water in the receiving tube may be less accurate. But nevertheless, distillation method is used for very low moisture containing food. On the other hand, we also use the Karl-Fischer titration. This is used for foods that show erratic results when by the oven method or the vacuum oven method. So we use the Karl-Fischer titration for many low moisture foods such as dry fruits, chocolate, roasted candy, oils and fat, or any low moisture food which is high in sugar or protein. So this is a titration method which involves using the reduction of iodine by sulfur dioxide, which uses 2 molecules of water. So in the titrator, you have the iodine solution, you have the sulfur dioxide solution in appropriate form, that is 1 mole of this. It is added to the sample and then titrated. The sample is titrated with the solvent, and once the iodine is consumed by the amount of water, the excess of iodine will not react with the water and the end point will be the color of iodine. So here you have iodine which is used as the endpoint. So this is the equation which shows that iodine and sulfur can react in the presence of water to form C5H2SO4. So there are 4 methods by which you can determine moisture. That is the oven drying method, convection, vacuum oven, the distillation, and the Karl-Fischer titration. This is the picture of an automated Karl-Fischer titrator. So you have the solution containing the iodine and sulfur dioxide. Now coming to water activity. What is water activity? Actually, the water activity of the food is a better indicator of the perishability or the spoiled nature of the food. And it is generally used to predict the shelf life of the product. Water activity is defined as the ratio of the vapor pressure of the substrate or the food to the vapor pressure of pure water at the same temperature. So AW is equal to P by P naught, where P is equal to the partial pressure of water in the sample, and P naught is the vapor pressure of pure water at the same temperature. So you don't need to measure the partial pressures because usually a manometer is used. We have got what are called as water activity meters. Just like a pH meter, you have a water activity meter which will measure the water activity. But water activity is generally represented by the term ERH, which is the equilibrium relative humidity surrounding the product. So this term the equilibrium relative humidity is equal to 100 into the water activity, or water activity is ERH divided by 100. So it is defined as the RH of the air surrounding the food, at which the product neither gains nor loses its natural moisture and is in equilibrium with the environment. So you can measure AW or ERH using a water activity meter. So as I indicated, please remember water activity and moisture content are two entirely different entities. So water activity, if you look at jams, they have got a high moisture content but a very low activity. Now you will ask the question, how is that when it has got high moisture content and low water activity? This water which is present in jams is all bound to the sugar which you add to the jams, and so therefore this water will not be available for microorganisms to grow. So foods with low water activity cannot support microbial growth, and we will see in a table pathogenic and spoilage bacteria do not grow in food with AW which is less than 0.5. So the water activity can be altered by food processing techniques such as drying, concentration, adding salt or sugar to reduce the water activity. So here is a table which gives you the water activity. So you see the water activity of distilled water is taken as 1 and the others are compared. So if you look at the top portion, the water activity is very high, and these are the foods that are prone to spoilage. So you see where you have very high water activity, so these are prone to spoilage. Whereas you have low water activity in biscuit, noodles, dry vegetables, soups, pasta. So these are not prone to spoilage by micro organisms, but to food spoilage by microorganisms. So low water activity is what is preferred for food preservation. So water activity and moisture are related by what is known as the adsorption and desorption isotherm. And you will see that the water activity is related in a non-linear relationship to the moisture, and this is known as the isotherm. So the uptake of water is termed as adsorption, and the loss of water is termed as desorption, and this provides a record of the water activity of the food at a particular temperature. So you will see this is not proportional to the moisture content, but you see as the water activity increases, you will see also moisture content increases, and with moisture content increasing, you will see the spoilage is there. Here is a water activity curve which is used to predict the safety and stability with respect to microbial growth due to and then any spoilage due to chemical spoilage, biochemical reactions, and due to the physical properties. So this water activity stability thing, you have the water activity here and you have the moisture concentration. What you need to see here is if you see only at water activities of about 0.85 or 0.86 and more, the pathogenic bacteria or the spoilage bacteria go. But however, yeast can grow at about 0.78 to 0.8. So you will have yeast and mold. So if you look at the water activity of bread, the water activity of bread is between this, and so you see bread is very prone to yeast. Whereas mold growth can grow at much lower water activity, that is 0.6, you will see that mold growth starts and it will increase as the water activity increases. You also see that the enzyme activity, that means spoilage of the food, due to enzyme activity starts from about 0.1, and as the moisture or water activity increases, you will see that there will be spoilage due to enzyme activity. So also due to lipid oxidation. So this line that is drawn over here is beyond 0.86 or 8, between 0.6 and 0.8. This is potentially hazardous food, and water activity below that are non potential hazardous foods. So the last slide here. So the last slide shows you the relationship of water activity and which bacteria grow. This is very important when you are designing a shelf life study. So you see at very high water activity nearing that of water, Clostridium botulinum, Pseudomonas, E. coli, Clostridium and Pseudomonas. At about 0.95, you have E. coli, Clostridium perfringens, Salmonella, and Vibrio. 0.94, you have Clostridium botulinum A and B and Vibrio. And you have Bacillus, Listeria, Bacillus. So above zero point 0.9 is potentially hazardous, which permits the growth of this. So therefore, water activity is used as a measure to predict the spoilage of food. Thank you.